All right, so why don't we get into it? The next speaker is David Whyte, who is going to talk to us about how luminescent fungi glow in the dark. So over to you, David.
Got it all. Well, thank you all for, like you say, coming back after morning tea. I thought that I would do after the, I guess, interesting discussions we've had at previous forays around luminescent fungi. I was intrigued to know how they actually worked and why they luminesced. So this talk is hopefully going to answer those questions, even though I'll let you in with a heads up. It creates more questions than it answers, as good science does. I would like to point out this is Joseph's photo from either the last foray or just after the last foray in Stewart Island, and was totally stoked to see that on Wikipedia it's the image that's used right at the top for the list of the bioluminescent fungi. So I thought it was pretty exciting that work that was done down here in New Zealand was right at the top of the Wikipedia page. Anyway, a very brief overview, because it's already been covered, I guess, in previous talks about bioluminescent fungi. Apparently there's 71 fungal species, or 106 if you believe Wikipedia. I think Wikipedia counts mycelial glowing or bioluminescence as a species, whereas I wonder if the 71 fungi species talked about in the scientific literature just talk about the actual fruiting bodies, but I don't know. And of those, obviously, as you've read already, 52 are mycelial. 12 of them are the jack-o'-lantern types, and the 5 Armillaria, which we get in New Zealand. And I have no idea about the 2, whatever that Latin name is. I'm not going to butcher that. People who can read it, but I'm not too sure where they fit into the scheme of things. But what is really quite fascinating is that they're all wood decomposers. They're all from areas of high humidity and warm climate. I'm not too sure what warm climate is defined as, but I think that's in terms of, I think New Zealand as a whole is termed warm. And they all digest lignin. So they all are a white rot fungus. And it's a bit hard to know where to pitch things, especially on Zoom. But for those who are not familiar with what makes up wood, if you have a look at this picture here, the cellulose you can think of as being the steel bars, and the concrete is the lignin, which holds the steel bars together. For those who are a more scientific definition, you've got your hemocellulose as your bars and your lignins that hold these bars together in the wood. So the white rot fungus digests the brown lignins and leaves the white hemicellulose there. So it turns out that the bioluminescence is driven by a luciferase. You can see Latin's not my strong point. Reaction, which is what I would consider the standard bioluminescent process that occurs across lots of different, I guess, species, worms, insects, other type of biological systems. And so your famous fireflies and glowworms in New Zealand use this system where you have your substrate, which is your luciferium, and then it's oxidized enzymatically by using an enzyme that's called a luciferase, and then you end up with this oxidized luciferin and light energy put out. Obviously, well, it's named because of Lucifer being the name for light production or fire production back in the day. So that's the general model that works across all insects and all life forms of bioluminescence. In terms of fungi, it's a wee bit more complicated. So below the line there, if you're a biochemist, you can read this. If you're not a biochemist, just ignore below the line. I'm not actually going to explain below the line, but I came across it and I can vaguely follow it. So I've left it in those for who are biochemists. But the rest of us who are not biochemists and don't want that level of detail, we'll just focus at the top here. So the fungal bioluminescence is a multi-stage process. So you've got your molecule of luciferin here, which is combined with NAD and hydrogen to produce the 2 hydrogens on the luciferin, and your NAD is changed. Is it oxidized? One or the other change. And then this molecule here is then oxidized using the luciferase enzyme, produce an oxidized luciferin plus water plus your quanta of energy, your light produced. And so it's a wee bit more complicated than the standard one that we've seen in the previous page. Different fungi have different Ls and leaf phases as well. So depending on the fungi type will depend on your specific molecule structure of your leaf-cicerin or your leaf arrays. And this is what it actually shows down below, but I'm not going to explain that because it's out of my league. So looking at some examples here, this is the Mycena from Australia. As you can see, it is obviously glowing at night with bioluminescence. And so they did some work on the gills of this one, and they found that the luciferin was actually vitamin B2 or riboflavin, which had been had a monophosphate body added to it, or this flavin, I'm not going to even try and pronounce that one. They couldn't isolate what exactly molecule it was, but when you stripped out these molecules from the gills, then you got no bioluminescence from this Mycena. So this is obviously operating as the luciferin, which kind of makes intuitive sense because vitamin B2 is actually a fluorescent molecule. So if you take a drop of vitamin B2 and drop it into a flask and then shine a UV light on it, then you get this wonderful green glow. And it's also why your energy drinks look a bit green in the light, because if you've got a water standard and then you've got riboflavin standards, which is your B2, your Rockstar or your Monster energy drinks also fluoresce green because of vitamin B2 in it. And when you drink them, the vitamin B2 floods your system, recharges you, and then gets excreted out in your pee. So your pee turns bright yellow, which is the riboflavin or vitamin B2. And then that will fluoresce as well, if you really want to know that. And milk will fluoresce as well. There'll be a green fluorescent in milk because milk has got a reasonable amount of vitamin B2 in it as well. So that glow looks very similar to what you'd see on a fungi glowing. So it kind of makes intuitive sense maybe that it's used in the process. But the big question I have is why? Why would you put the energy into glowing? And so this study, which used this neo something, which is a tropical fungi found in the forests of Brazil and other tropical areas, created this model here where they attached an LED, and a green LED, into a fungi, plastic fungi fruit bottle, and then connected it to, not milk bottle, plastic fruiting body, connected it to a battery, and this is what it then looked like at night time. And as you've probably already read here, the LED, which has got this nice beautiful peak at 525 nanometers, has an approximate same spectra as the green glow that comes off these fruiting bodies. And so you can see in these results here that it attracted, and these are significant, so the ones that are not starred are the significant ones. So a significant number of these types of beetles, and then a significant number more of flies. The blue ones are the controls. And so no of these beetles on the controls, small number of flies, none from this type of beetle, and a small number from here. And these were almost statistically significant. So they were just off statistical significance. And so the idea hypothesized behind this is that being in a damp environment and a humid environment, and these things fruit close to the forest floor where there's not air movement, that the insects could be attracted to them, get spores, and then disperse them as spores, since the wind is not necessarily going to disperse them well into the environment. So that is one theory behind why they actually do glow that seems to be supported. And another example of why this is supported is this Mycena here, which is from, what's next to Spain? Portugal. Has a Cardian clock. That is, it switches off during the day, so it doesn't glow during the day, and it switches on at night. And so therefore the fungal organism is clearly turning it on and turning it off, which would imply that it wants to glow. It's just not a function of, say, it's being alive. And so therefore there's an active part in attracting these insects. So that kind of makes sense, except that any good science, you get more data which confuses things. So this is the Australian data. So this is one of the jack-o'-lantern species from Australia. That's a native one, and it fruits at the base of eucalyptus trees, and it fruits in midwinter. So in June, and then it has this fantastic glow that's associated with it. And so these folks split up a number of fungi that were glowing, spread them around the eucalyptus forest, far enough away from the light sources, etc., and produce, put these yellow sticky things that are used actually to control bugs, flying bugs in greenhouses, and put them near or right next to the glowing bodies to see what stuck, and then did the analysis. And the results here, the control actually on average got more bugs stuck next to them, which were they didn't have any fungus next door to them in the controls, whereas the glowing fungus actually got slightly less. Obviously no statistical difference, but I would draw your attention to the number per trap per night. So this is around half a insect per night. So over 2 nights you get 1 insect, whereas the fungus glowing only attracted 1/3 of an insect every night. So that's 1 every 3 nights. So the other key thing in this is that this fungus actually glows both day and night. So it doesn't switch off at night time, it's just on all the time. And it's fruiting when there's no flying insects about, because we're in the middle of winter. And so therefore it doesn't seem to attract more flying insects. They didn't look at beetles or crawling insects, but it kind of makes sense that with no Cardian rhythm and the time it's fruiting, that for this glow-in-the-dark fungi or bioluminescent fungi, there is no insect attraction occurring. So mycelium also glows. So this picture here is the cultured mycelium, and you can see in the dark here that it has got bioluminescence occurring. This is the fruiting body of this fungi, and the fruiting body while it's bioluminescing. So mycelium seems to be quite common, if not more common, than fruiting bodies glowing. And so why would that be the case? Which is an interesting question. The mycelium of what we've just looked at the previous page, if you take it into and put it into the lab, it does have a 22-hour rhythm to it. So at 21 degrees Celsius, if you hold it there, you can see that you've got a minima, and then another minima, maximum minima, maximum minima on a 23-hour cycle. And then at different temperatures, you get the same effect, sometimes not as obvious. And so I think this, the paper didn't say this explicitly, but I think this on the right-hand side is actually a Fourier transform of the data to show where the peaks and the troughs are. So there's obviously some kind of rhythmic approach to mycelium, which would imply that there's some kind of control or some kind of effect going on. It's just not glowing constantly. If you put the mycelium out into the areas where mycena grow in, the subtropical warm areas, they will attract insects, but that doesn't seem to be logical given that mycelium is in the ground, and so therefore you're not going to attract flying insects to it because you can't see it. But people have hypothesized, but there is no data out there, no studies done, that you could be repelling things. So some fungi show effects of light, so maybe the mycelium is glowing so it repels other fungi so it doesn't get attacked. It might be repelling fungosaurs or protasia, which might attack the fungi, but no one has looked into it, so that's purely conjecture or hypothesis. But the other hypothesis is that it could just be a product of growth. So there is some kind of relationship between growth and luminescence. It's not a direct relationship, but it is some kind of a causal relationship. And so there is an optimal temperature for light output, which seems to be roughly related to the optimal temperature growth, and same with the optimal pH. So as you change the pH away from the optimal, then you get a change in light output and a decrease. And that seems to be roughly correlated to the optimal pH for growth. And the other thing is it stops with toxicity. So heavy metal toxicity will stop the fungal mycelium glowing. And they've actually used that to produce a bioassay for heavy metals. And so you cultivate up your mycelium, again, using this fungi at the top here, and then you set it away and incubate it up and measure your light output to start with, and then you expose it to your solution that you'd like to expose it to, incubate it through the process, and then measure it again with the luminometer, and then compare your final with your initial, and then you get your inhibition, and from that you can back calculate your heavy metal toxicity, which apparently is a good and inexpensive way of doing this, instead of sending things to a lab to try and work out the heavy metal. Now, So in terms of future, there's some interesting things that while we're out fungal hunting in the bush, we might like to keep an eye out for. And one I would call the high light intensity fungi. So in the older reports of this Australian jack-o'-lantern fungus, people were able to see it from 1,000 paces away, which I presume is close to a kilometre away. And that obviously must be fairly open eucalyptus land. And more recently, people have talked about seeing it in the scientific literature 40 metres away, and being able to read a newspaper under it as well in the old literature. And so that's pretty remarkable and pretty glowing. I've not heard of any reports in New Zealand that show that intensity. And most fungi seem to be fairly low intensity, where they talk about 15 minutes having to be in complete darkness to adjust your eyes in the older literature to see it. So there could be in New Zealand some ultra bright fungi, and maybe we could discover that. The other thing which I find really, really interesting is currently all known bioluminescence appears to be green, and so their peak wavelength is somewhere between 520 and 230 nanometers. Whereas when the old literature, I'm saying 1924 here, people were talking about blue, green, yellow, and white bioluminescent fungi. And of course, being older literature, we don't know if what someone was saying was green was actually blue, and vice versa. And so it's very hard to know. And currently, we've not found any. But this is quite an interesting paper where we've got, this is our luciferin molecule. And so this molecule here, R equals 1, this is what is on the luciferin molecule to start with. But what you can do is attach numbers 3 to 9 to the molecule, and you get a different light colored output. And so, for example, here we numbers 6, 7, and 3 are bluer or greener, and number 9 is redder. And so there are the technical possibilities that there are different colors of bioluminescent fungi. In terms of C and D, C is the efficiency of the bioluminescence. So this one here is the standard fungal bioluminescence that exists already. So there is a green and a bluer, greener and a bluer color that is more efficient. Well, is the same efficiency or more efficient. And so therefore, again, that's technically showing that there is a possibility that we might actually see blue luminescence or greener luminescence compared to the standard. This one down here is the extinction coefficient, which is a wee bit above my pay grade, except I would say that again, the one here, which is the standard one found in nature that we know about, let's say, for example, 7 is within cooey of that as well. So again, showing there's possibilities that we could actually see different spectrum of the bioluminescence. And if that was the case, that would be a world first in my understanding. So I think I will stop sharing. I'm happy to take any questions. I have gone faster than expected. No, I haven't actually.
No, you're just right. Excellent. Thank you. That's really interesting. It's amazing. Yeah, I collected Omphalodus netiformis in Australia once when I worked at Melbourne Uni, and we took it into the dark room, and within about a minute of worrying that you couldn't see much, it was like, whoa, the whole room's lighting up. It was amazing, actually. Great. Has anyone got maybe one question? We have a bit of time there. Yeah, 1 minute. No.
All good.
All right. Thank you. Really interesting. Oh, hang on. Anna's got a question. Okay, you can keep typing, or do you want me to let you ask it yourself, Anna? I can do that. All right, Anna, you should be able to make yourself unmuted now.
Did I? Am I hearable? Okay.
You are.
Hi, David and Bevan. I was just wondering, David, why when mycelium and just the mycelium glows, which in some species there's not even any part of the mushroom at all that glows, when it's just mycelium, why wouldn't we consider it possible that attracting insects that bore wood could be a reason for that glow? Because could they not disperse by propagation rather than by propagation of the mycelium itself rather than by spore dispersal? Or is that just not something we think that would be a strategy of the fungi?
I didn't read that in the literature review, but that's a very good point. And yeah, I mean, I think that's an excellent point. So yeah, I don't know. And again, it's a big unknown in this area. So.
Thank you.
Right. Okay, thank you very much, David. Really good. So I'll leave it there.